I'm going to say a few words about her, and I'm going to start off kind of on the very academic side. This is going to be a fairly brief introduction, and then I'm going to say some things about her more practically in terms of what she does to have impact on people beyond her research. Karen is an associate professor in our speech language pathology program, and she's also in our neuroscience and education program, two programs that sit side by side in this department, biobehavioral sciences. So she's in the neuroscience and education program and also our speech language pathology program, and those two programs are mutually informing at this stage in many ways. She got her doctorate in the United Kingdom and did a lot of work in theoretical linguistics as well as speech pathology. And was a postdoctoral research fellow at the Center for Developmental Language Disorders and Cognitive Neuroscience at the University College London. So she's been in one of the great research centers in the world doing this work where she started. She participated in the development of an important screening test for grammar and phonology. So although she's working in neuroscience, she's also really thinking about how you can screen for disorders and then what kinds of assistance you can provide to people in improving their performance in speech. She was a research fellow early in the last decade at the Mind Articulation Project at MIT, which was one of the leading efforts in the United States to bring together the new cognitive and neuroscience work with the activities of mind in speech and other areas of performance. But when she came to Teachers College, she founded the Neurocognition of Language Lab, which is an operating laboratory for the study of the kinds of things that she's going to be talking about tonight. And you're going to see some of her students. I think you're going to see a little bit of her equipment from that lab. And she's been really heroically doing that work, which is scientific work. It's also educational work. And it's drawing graduate students into these projects very deeply. She collaborates with the neuroscience folks at the medical center and at Columbia Arts and Science. So there's a big community of people here at Columbia doing these kinds of work. I want to talk a little bit more about the practical side just for a minute, that she's a person who really wants to take this kind of work, which is the most advanced work of its kind being done in science and education and development of curricula and things like that. She wants to take that to places where it really makes a difference. And one of her projects is taking graduate students with her to Cambodia, one of the poorest countries in the world. A place that just lacks infrastructure and lacks the economic resources to do very much in terms of institution building. She has been going there with her students and developing networks of rural healthcare and screening and helping to develop capacity for the work in her area. And along the way, she's been exploring the concept of telepractice, which is how you can take the dense, advanced resources in a first world research center like this one and make them available on the ground in places where resources are lacking. And so she's really a visionary in trying to cross over and make available to everyone the kind of work that would otherwise only be available to some. So with that, I'll introduce Karen Froud.
I want to thank you all for coming this evening, and special thanks to Provost James and his office for inviting me to speak to you tonight. The title of my talk is The Neuroscience of Language and Learning. That's a very big title, and I think we can't really talk about that. We have maybe half an hour together. So I narrowed it down a little bit in my subtitle, which is the work of the Neurocognition of Language Lab at Teachers College. But even that is a little misleading. We do a lot of work in my lab, and I will tell you about some of it. So we're going to talk about a little corner of the neuroscience of language and learning that's in a little corner of the work that my lab does. So it's rather more modest than the press releases would lead you to think. I want to talk about not just the work that my lab does, but also some of the implications that we think that work has within a neuroscience and education framework. And I do want to mention before we start that I have here around the place, planted strategically, various doctoral and postdoctoral and other collaborators who've done work in the lab, and all of them would be happy to talk to you about their studies or about any of the other work that we're doing. So I do hope you'll stick around afterwards as well. Okay, I'd like to really start by pointing out that Teachers College is really unique in this respect in terms of having the expertise and access to this kind of research right here, integrated within a school of education. Even though I believe that neuroscience has a great deal to say to education and vice versa, for sure, it's not typical for institutions that specialize in teacher preparation to have direct access to these kinds of research. And because we do have this expertise and this facility, I think there are tremendous opportunities here for innovative research in education and neuroscience that might not be possible in other institutions. Here are some pictures of our lab, well, little corners of the lab. And I really want to point out that we do have a very special environment. It allows us to really control the circumstances under which we carry out our experiments. We have very special equipment, and it needs great care and attention and expertise to keep it running smoothly. We did bring down some of our equipment tonight, but this is not the typical environment for carrying out this kind of work, so we'll see how that goes. In our lab, we carry out brain imaging studies that give us detailed information about how very rapid processes in the brain are unfolding over time. Now, most people, when they hear the term brain imaging, they're thinking about functional MRI images, right? Those beautiful pictures of brains showing where things light up when a certain mental process is occurring. And people think of this like taking pictures of the brain, and in a way that's what we're doing, but let me explain that what we're really taking pictures of isn't brains, but activation inside brains. In the case of functional MRI, the images really show where in the brain more rapid rates of oxygen uptake are happening. And using that information, we can get really cool pictures that show us where in the brain some activation happened. But because brain processes happen so fast, by the time oxygen levels change, the activation that caused that change is already over. So we know that the sequencing of events is very important for the brain. And we know that when we look at where things happen, we're missing information about how quickly those things happened. So we need to use other imaging techniques that can tell us about when things happened in the brain. And that's what the kind of brain imaging that we do is really good for. We can do this by measuring the actual communication between brain cells. When brain cells talk to each other, they generate tiny electrical fields. And that's the kind of measurement that we can make. It's called EEG, or electroencephalography. So people who work in education, and I know that's many people in the room tonight, either directly or indirectly, teachers, psychologists, researchers, scientists, they've been doing great work for decades or even centuries now. And really, neuroscience hasn't been part of that picture until relatively recently, let's say maybe the last decade, maybe a little longer. So one question I often get asked is, why should we pay attention to this, right? Why neuroscience? Well, in response to that, let me point out that anytime we want to understand something about learning, we must understand something about the brain. The brain is the source of all our behavior and experience. And by now we know this is not a one-way process. The brain is not some passive receptor of information. Brain imaging studies have shown us that even if we're processing some very simple piece of sensory information, we're not doing it in a passive way. We're actually creating our own experiences from within, literally moment by moment. So taking in information, any kind of learning experience, is far from a one-way exchange. And we need to understand how that works, how information is being processed as it's received, and what is the role of our own pre-existing experience as we learn and as we acquire new knowledge. But there's another part to this too. We do understand that some behaviors that we can observe on the surface are actually a lot more complex than they seem. So think about reading, just as an example. What is happening when you show a child a written word and you ask them to read it. So you show them cat written down, and they say, that says cat, right? It seems very simple. We can observe the behavior. We can see if the child reads the word or if they don't, right? We can observe whether the child makes a mistake. And then if they do, we can use that information to infer something about what process might have gone wrong in their reading. But if we only observe the behavior, we can't directly see the relative contributions of all the different kinds of knowledge that have to go into that process. So for instance, the child has to know something about the sound system, the phonology of the language that they speak. They have to know something about the words, the vocabulary of their language. They have to know something about the writing system that's used in their community. They have to know how you can map between a set of visual symbols, right? Marks on a page, it's very abstract, and a set of sounds. Right? And between that set of sounds and a word, and then between that word and some concept, some idea of an object or an action that's encoded in that word and that maps onto something in the real world. Also, the child has to be able to direct attention, cognitive resources, sensory resources appropriately, and they have to be able to sequence speech sounds appropriately. And no doubt there are many other steps in that process too. It seems so simple from the outside, but we know there's a lot more to it. I was talking to a colleague of mine earlier today about this issue, and he likes to use the analogy of a car. He says if you're interested in cars, you can look at cars as they drive past. You can say, that car goes fast, or that car runs smoothly. If you're really, really interested in cars, you can get into a car and figure out how to make it go fast, how to make it run smoothly. But unless you open the hood, you can't see what the components are that allow it to go fast or run smoothly. And really, even if you open the hood, you're still only looking at some things that seem to be related, right? You need to make more detailed observations than that. You need to know there's something electrical over here, some water over here, some gasoline over here, right? You need to know how those things interact and what is the sequence of those interactions. And that's when you start to understand something about how that car can move, right? So I think this is a useful analogy. If we only look at behavior, we run the risk of missing a lot of information about the processes that must happen in order for that behavior to take place. We might not understand, for instance, some things about why the behavior might be difficult for some person to learn when it's easy for other people. We might not know about underlying individual differences, about how one person processes information that's different from how another person does it. Technological advances in the past number of years have meant that these kinds of questions are much more accessible now than they have been before, using neuroscientific approaches. But let's be clear about why this is useful. If we can learn about how people learn, we can better inform our teaching approaches. We can help to provide teachers with tools for more effective learning. We can provide clinicians with better tools for intervention. We can begin to evaluate our own work, our clinical approaches, our educational approaches, at a more fine-grained level of detail and with greater objectivity than we were able to before. And this applies not just in classrooms, not just to childhood learning, but across the lifespan. So let me take just a little time to explain something about the technology, how we actually do this. In fact, there are many different possible approaches, but the one that I'll describe here is a methodology that we call the event-related potential approach. Now, we're lucky to have with us tonight a real live model. This is Heather Green. She is one of our doctoral students from the lab, and she's kindly agreed to be our guinea pig for tonight. We also have a couple of other doctoral students. This is Shyle Maddox and Trey Avery. Trey is also my lab manager. And so they're going to come and show us how this actually happens. We brought, as I mentioned, some of the lab equipment down here. And I do just want to keep reminding you, this is not how we do research, right? With a big audience in a big room with candles and lights and things like this. We need a much more controlled environment than this. But it will give you an idea of how this works. So, oh, wait. I kept doing that today. There we go. First of all, because we're measuring communication between brain cells, which is basically carried out in the form of these tiny electrical currents. First, we place, as you can see, some electrodes on the head. Then, because we're interested in how people process information that they receive, we have a computer that can present stimuli to our participant. We have to do this by computer because, remember, we're concerned here with how very rapid brain processes unfold. If we're going to know when precisely something happened in the brain, then we also need to know when precisely something happened in the environment that the brain is responding to. So when the person heard a sound or when they saw a word, for instance. So for this method, we can't do things like showing someone a picture or holding up a flashcard or putting someone in a classroom and see what happens, because we need to know to the millisecond when something happens. So it all has to be done by computers. And also, I should say, not just any computers, because computers don't really like to talk to each other that fast. So we have to get special computers, and we have to keep them talking to each other in a very kind of specialized, fine temporal way. Okay, so electrodes on the scalp get connected to an amplifier. And this amplifier, this is the really important piece of electronic equipment that we need, because the signals we're recording are so very, very small. The amplifier is very specialized and really sensitive. That's actually the most expensive and important part of this setup, apart from the brain inside this head, right? That's, yeah. So the amplifier then is connected to a computer that records data about the electrical fields that are being generated. But wait, let's look at the, here we go. This is going to be awesome. There we are. Okay, so what you can see here, each of these wiggly lines is one of these electrodes that's on the scalp of our participant, right? And the reason that these lines are going up and down is because the communication between the brain cells is happening continuously. Right? So what we see is voltage fluctuations as those electrical currents change. So some of what we record, because this equipment is so sensitive, and this is a noisy environment, is from the environment itself. Right? So it makes a difference to us, for instance, that the lights are on, or that there's mains electricity running through the circuits in this room. So the equipment is that sensitive that you can see. Let me show you a couple of other things. In one moment as our recording line goes past. Okay, Heather, can you blink 3 times, please? Okay, so now that's an eye blink, right? So an eye blink is a very, very small movement, but you can see that that's so much bigger than the kinds of signals that we're actually recording, right? We're recording really minute signals. Heather, nod your head for us, please. Right? So again, these very small movements, what it's recording there is not just the change in location of the electrodes, but actually the electrical communication between nerve cells and muscles that allow you to do this, right? But those electrical communications are much, much bigger than the ones that you get in the brain. Okay, so the ones that we're trying to record are very, very small indeed. Okay, well, let me switch back. And Heather, thank you. You're free now. After we've recorded some data, kind of looks like this, right? That's really a little snapshot of what we were looking at just then, but in real time. We have to do some more things to it to get some information out. So in this data stream, can you see the wiggly lines there that are for each electrode, right? But then if you look up at the top, do you see those little square things? Yeah? Those are little, we call them flags because they look like flags. So yeah, we're an imaginative bunch. But those little flags actually get sent to our data acquisition stream by the computer that's presenting stimuli, right? And that's how we know exactly when something was presented to the participant. Okay, so then what we do is we chop up our data. We look at the brain's responses to those particular events, right? And then once we have those segments of data, we can average those segments together, and that's when we get what we call an event-related potential. Now, what averaging does is, basically it makes the signal-to-noise ratio better. It means that all the stuff the brain is doing that isn't related to processing the stimulus, like it keeps your heart beating, right? That's important. We don't want it to stop doing that. You monitor the environment all the time, and you make changes internally to compensate for how the environment is. I don't know about you, but I'm finding it hot in here. Is that, yeah? Could be just me. Holding your body position. Thinking about that cocktail that you get to have in a while after this talk is over, right? Like all of that stuff. The brain is continuously doing that, but that's not related to our question of interest, which is how does the brain process that stimulus, right? So if we want that information that's related to the event that we're controlling, then what we do is average together the brain's responses to every time that event is presented, and that's when we get the event-related potential, right? We call them ERPs, right? ERPs are really like signatures of brain processes. The brain activity patterns that are typical for processing a particular kind of stimulus. Okay, so on this slide, we're looking at some ERPs, okay? Remember, these are the typically occurring brain activation patterns that follow some stimulus. And the stimulus could be a word or a sound or something that you see, some dots or a picture or something like that. Here, these pictures of ERPs have been organized along a timeline that starts when some stimulus like that is presented. An early ERP, one that happens very soon in the processing stream, is one that we call MMN. It stands for mismatch negativity, but there's no test later, so don't worry. MMN is just its name. We can call it Fred or Bob or whatever you like. But you can see that the MMN happens around 100 milliseconds. A millisecond is 1/1000th of a second, right? That's the kind of time scale that we're looking at within the brain. In this picture, you can see that there's a pattern of voltage seen with MMN, a negative kind of voltage charge. Can I do this? Yeah, you see that. This kind of blue patch here means that the voltage actually takes a dip. It goes a little bit more negative than the baseline state, okay? And that means that the MMN has this particular kind of unique signature. It's actually generated by auditory cortex. So when you hear a speech sound, your auditory cortex about here, right, on both sides, generates this change in voltage that then you can map at the top of the scalp, okay? So it's timing and it's a pattern of activation. That's how we identify an ERP. With MMN, it's known to index something like our ability to recognize differences between speech sounds. So if you hear two speech sounds in your native language, MMN. If you hear two speech sounds not from your native language, no MMN, right? So it's an interesting ERP, actually. So MMN about recognizing speech sounds. Then a little bit later on, we have something called an N170. It's another brain signature. N170 is associated with automaticity of processing or expertise. Not being an expert in something, but just, something that you can be really fast at, something that you can do really automatically. We see it in people who are really good at recognizing something. Like some people are really expert in cars. Cars keep coming up tonight. There we are. It's a car night. So some people are really good at recognizing cars, and then we see MMNs, N170s, when they see cars. But for most of us, it's something like, reading. If you're an automatic reader, if you're someone who learned to read as a child, then you will show N170 responses when you're reading, because that's something that you do automatically and very fast, okay? The N400 is next in our timeline. N400 happens about 400 milliseconds after you see something. N400 is a very interesting ERP. It seems to index our knowledge of meaning, and mostly it's been investigated with respect to linguistic meaning, but we'll talk about it some more in a minute. And then P600, that's the last one we'll talk about today, even though, I picked a little subset. There are a lot more of these ERPs that we work with and that we can discuss further. P600 is another ERP that has been mostly investigated with respect to language, but in this case, it's about our knowledge of structure, the rules that govern language, like syntax, right? Grammatical structure. So what I'd like to do now, just for the next few moments, is talk about some of the current studies happening in our lab and tell you more about each of these ERP signatures and how we're using this kind of information to help us answer some interesting and useful questions. So we'll start with our friend MMN, right? Or Fred or Bob or whatever we decided. So, in a study we just recently published, that's myself and my colleague, Professor Khamis Dakwar, who's been a long-time collaborator of mine. We focused on this speech disorder that's called apraxia of speech. Children with this speech sound disorder have been thought to have a disorder affecting the motor movements needed to produce speech. Speech is very fast, especially when it's me doing it, right? I talk fast. I talk a lot, so I have to go quickly. So you have to be very coordinated to get all of these movements in the right order. We take it for granted, but it's very complex. It's very difficult. And the idea has been that children with CAS, that's childhood apraxia of speech, that they have a problem doing that, coordinating those movements. They're just not very good at that. But CAS has been very slow to respond to therapy that tries to address these problems, and really nobody knows why. We keep trying, but we're not really moving forward with that. So recently, we looked to see whether children with apraxia of speech are really processing speech sounds the same way as their unaffected peers. We can elicit an MMN response in an unaffected child quite easily. What we do is this. We play a sequence of speech sounds like this. Are you ready? Okay, you're going to be in an experiment now, okay? All right. Pa, 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 ba, pa, 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 ba, pa. Okay, that'll do. Who heard two speech sounds? Okay, anybody hear only one? Okay, anybody hear more than two? It's possible. I aspirate things. It's the accent. It's a problem. It's a difference, not a disorder. But all those of you who heard two speech sounds there, when you heard the one that didn't match, right, the less frequent one, your brain gave an MMN response, right? I guarantee it. Even though we weren't measuring, that's what happened. Now, of course, in order to see that, we would have to average together your brain's response to all the pars, and then average together your brain's response to all the bars, and then compare the two, right? And then we'd be able to see that there's this difference. Okay, but that's how we get an MMN. So, in children with CAS, guess what we found? No MMN. It's not there. In comparison to their unaffected peers. So really, this shows that children with CAS are not processing speech sounds the same way. It's not just about the motor movements, although that could be a related issue, and it probably is. But it's also about the representation in the brain of the speech sounds that they're hearing, right? So what this is groundwork that takes us a step closer to understanding this disorder. In the future, as we're building capacity in the lab and undertaking these more in-depth kinds of investigations, we really want to build on these kinds of results. We can start to ask questions like these. Can we differentially diagnose disorders like this more effectively? For instance, perhaps some children with this diagnosis really do have a purely motor speech problem. And for them, motor speech kinds of investigations might help. But some children definitely have this kind of problem that affects representation of speech sounds too. And for them, we need to approach intervention differently, right? We need to take a different tack. So we could also develop new and more appropriate interventions for children with particular kinds of speech disorders. And if we have access to tools like this that let us look below the surface at what's happening, then we can also see whether our interventions are really helping. Like, can we see MMNs start to appear in children who didn't have them before, right? This could give us a more objective means to evaluate whether our interventions are really working or not. Okay, so now let's move along our timeline a little bit and take a look at this N170 component. Remember, N170 is the brain activation pattern that we can associate with automaticity or expertise, right, in a particular domain. This study is one that one of the doctoral students in my lab is working on, Laura Sanchez. She wasn't able to be here tonight. She has a beautiful toddler that she had to be home with. But Laura was a literacy specialist in the Dominican Republic before she came to us. And she knows that adults who attend a literacy course do not necessarily become expert readers, right? They don't necessarily learn how to read automatically. And if they don't, they're at greater risk of forgetting, in a sense, how to use this new skill that they acquired. Now, let me give you some background. N170 can be generated by the brain in different ways. If you're showing a language-related kind of expertise, like automatic reading, you should have a left hemisphere N170, right? And that's because the left side of the brain, for most people, is the part of the brain where the language areas are, right? So if you're using language in a fast automatic way, you'll see N170s coming from over here. In other kinds of tasks where you might become expert, like visual recognition of some kind of thing, then you might see right hemisphere N170s, okay? So the brain can generate this same signature, but coming from different places. Now, for people who learn to read as children, we get left hemisphere N170s in any reading task. So down here at the bottom, we're showing two different reading tasks. In this one, you see some words, and sometimes you see a repeated word, okay? If you see a repeated word, you have to press the button. Now, notice that you don't actually have to read to do this task, okay? It's called a one-back paradigm. All you have to do is know that those symbols are the same as the symbols you saw before, right? So if you can't read that language, it doesn't matter. You still can do the task. But for people who are automatic readers, they can't help it. They read anyway. So you get left hemisphere N170s in that task too. For people who learn to read as adults, though, we don't. In that task, we get right hemisphere N170s. That shows us that people who learn to read as adults are good at recognizing symbols, but are not automatically reading them, right? In this task on the other side here, oh, there it is. What we have here is a forced reading task. What happens in this task is that you see a word, and then you hear a word. And your job is to say, yes, those two matched, or no, those two didn't match, okay? So if you see casa and you hear casa, then you say yes. But if you see cielo and you hear hoja, then you say no, okay? My accent is, again, in the way there, so bear with me. Now, in this task, you have to be able to read the print word in order to perform well, okay? So for people who learn to read as children, no problem. Automatic reading, left hemisphere N170, no problem. For people who learn to read as adults, they may be able to do the task, but they show right hemisphere N170s. And what this indicates is that even in situations where they're trying to read, we see them using other strategies, more costly cognitive strategies, right? Something a little slower, less automatic, okay? So they're not able to tap into that rapid processing that the left hemisphere gives you for language. Okay, so here's where this could take us. As we move forward and we're developing the capacity to conduct, again, more in-depth investigations, we can start to evaluate the ways of teaching people that might help get them to that point of automaticity in reading. This could mean a lot of things, right? Maybe just more exposure, maybe different teaching methods, for instance, methods that emphasize decoding, for example. Maybe more practice, maybe faster practice, right? It could be a lot of different approaches. But since we have access to these methods that allow us to really tell when somebody acquires that automaticity, then notice that we can evaluate the effectiveness of these different teaching methods, and indeed the effectiveness of adult literacy programs, rather differently. You know, I'm sure you do know, people do take tests at the end of their literacy programs, and there's nothing really behaviorally that helps us to identify from those tests, this person managed to read automatically and this person didn't, right? People perform kind of within a range. Some do well, some do less well. But there's nothing about the behavior that really tells us this person is going to succeed, is going to hang on to those skills, and this person is going to need more input, more support. So this actually gives us a way to look at which are the people that might need more support to get to that point of automaticity. This is actually kind of a key point for us. Oftentimes we see situations where people are behaving one way, but when we look at the brain, we see that their brains are handling the same task very, very differently. In this situation, being able to see whether adults show a left-sided N170 response when reading could allow us to identify those who might be at risk of forgetting their new skill, which is something that the behavioral tasks really can't tell us yet. So that would be a useful application for this kind of work. Okay, let's talk about the N400 and P600 signatures. These are the last two in our little timeline, but remember, there are a lot more, right? I'm being very selective tonight. Now, N400 and P600 have mostly been investigated with respect to language processing, a gatecrasher. You know, when they come along, you've got to take them, right? We know that you can get an N400 when something happens with respect to linguistic meaning. So for instance, if we have a sentence that begins like this, he spread the bread with butter and socks. Okay, right. So everyone's expecting something like jam or jelly or marmalade or something like this, right? You get socks and your brain, you can almost feel it. Your brain goes, whoa, right? And that unexpectedness, that's when you get an N400, okay? So it's when there's a violation of the expected meaning, right? It's when you're expecting something to mean one thing and then you get some kind of surprise. That's when you get an N400. Okay. Now, if we change things up a little bit and instead of violating the meaning necessarily, we violate the grammatical rules. So maybe we put a verb there instead of a noun, for instance. So if we say he spread the bread with butter and walking, right? That's weird too, but weird in a different way. And that's when you get a P600, right? Because there, what you did was you messed up a rule, a syntactic rule, okay? Not the meaning as much as the structure. Does it make sense? Okay. Well, in a recent study from our lab, that's actually the study that Shail Maddox, who is somewhere over here, recently successfully defended to become Dr. Maddox, of which we're very proud, yes? She's going to kill me for that later. We actually found that you can get these brain signatures in response to other kinds of processing tasks too. And this is like really new information. It's very exciting. We set up a simple problem-solving task. It required people to make a decision. And we found that we could get N400 responses when people were using visual imagery, a kind of mental model sort of approach. But you get a P600 if people are using a mental rule or an algorithm to solve the problem, right? Cool, right? Yeah, that was pretty cool. So what it does, like, you know, all good research, is it generates more questions than we actually asked in the first place. So, for example, can we use this information to help us figure out the kinds of approaches that people prefer to use or default to when they're solving problems? People often describe themselves as things like, you know, oh, I'm a visual learner, or I'm a language-based learner, or something like this. Well, Is that really true, objectively, or is that just something that we say about ourselves, something we learn about ourselves? If we look at the brain, rather than asking people, we might be able to find out about that. We also know that things like problem solving, right, that's very complex. That's a really kind of big thing. It's not just one system that's involved. When you're solving a problem, you're thinking about all kinds of things. You draw on pre-existing knowledge, you use language, you use your previous experiences, right? All kinds of things go into that. So, of course, people use a combination of different approaches when they're trying to solve problems. So, in effect, we switch around between different problem-solving approaches. When we're using our brain imaging techniques, then maybe it would be possible to tell when and how those switches take place. And what can we do to support that kind of cognitive flexibility at different stages of learning and different kinds of skill acquisition, right? So it's opening up these new possibilities for investigation of how we learn, how we solve problems. Okay. So that was an extremely brief skate through some very kind of complex studies that have been developed over years. And as you can see, they're still in development, right? They draw on a very rich background from the research in neuroscience and psychology and education and linguistics and other disciplines too. But what I'd like to do now is really give a kind of summary of the sorts of questions that we're trying to address using these techniques that come from neuroscience. Now, when you think about what are the big questions in education today, what do you think they are? What is it we want to know more about? And what are the methods we can use to address those avenues of inquiry? I mean, I would say that at least one of those big questions really hasn't changed all that much in all the time we've been thinking about learning, thinking about teaching, thinking about education. I would say that what we really want to understand, what really informs all of our inquiry, is this drive to know more about how people learn, right? That's what we need to know. Now, we can refine that question a lot. We can approach it in different ways. We can ask related questions, like this one. Are the learning mechanisms the same across the lifespan, or do they change? Do children learn differently than adults? And if they do, what are the differences, right? How do we know? How can we tap into our knowledge about that? And then how can we use that information to enhance learning across the lifespan, right? Are the learning mechanisms the same for speakers of different languages? That's not to suggest, mind you, that someone who speaks English learns differently than someone who speaks American, right? No? Thank you. But when we think about people who are multilingual as opposed to people who are monolingual, which I really am, do they have access to different resources from one another? What about people who are really multilingual from early in life compared to those who learn another language much later in life? What are they tapping into? How is it the same? How is it different? And what about really complex sociolinguistic situations, like those that mean people always have to switch between different languages, different varieties of a language? Say where you speak one language, but you have to become literate in a different language, right? We've done some really interesting work on that too. But what we're interested in are what are the learning challenges there, right? What difference does it make? How are people handling that? And how can we support that? What about when we're teaching or learning different kinds of knowledge and skills? I talked a lot about language tonight because that's my personal focus. But that's obviously not the only learning domain of interest, not by a long way. So if we can identify learning mechanisms that help us in one domain and figure out new ways to tap into those, can we then extend that across domains? Can we figure out the most effective ways to teach across disciplines? Or is it really the case that we need to focus on each discipline in a different way in order to be maximally effective? Right? And how can we evaluate that? How can we look at this? Another major question that I think is important to many of us is what happens when people have problems learning? Why would one person be able to acquire knowledge and skills that are very difficult for another person to acquire? So think about reading again, just for a moment. When we see that someone, a child or an adult, has a problem learning to read, can we even begin to tell why that might be happening? Now, is it because they have a problem with language? If it's language, then is it the speech sounds? Is it the system of sounds and how they join together? Is it the vocabulary? Or is it the syntax? Can we even separate those things out right now? Could it be a problem with something else, like visual processing, or with being able to direct attention appropriately, or remember information that they already learned and make connections between those pieces of information? Or is it maybe because some people are just different kinds of learners and need to have information presented in different kinds of ways, right? We can use these approaches to help us disambiguate those questions. They are questions that are currently very difficult to answer. That's why they're the big questions, right? Generations of behavioral research have brought us a kind of accumulation of experience, some insights. But I'd suggest that maybe it's time to start asking those questions in different ways. Knowing that we could potentially have access to methods from neuroscience, maybe we can access a different level of understanding. Using these approaches that I've been outlining this evening really fast, and others like them, maybe we can move towards a more fine-grained, more effective evaluation, not only evaluating the nature of the learning challenges and the situations and the disorders that we might encounter, but also how effective our approaches are in those situations. Okay, so I wanted to close with a brief look at the principles that really govern the research program that we're developing in my lab. In this respect, this is my soapbox. I do believe that my lab is unique. And it's because we're carrying out neuroscientific investigations within a school of education. That's what allows us to approach our work in this way. By collecting scientific data from rigorously conducted experiments, we can start to shed light on questions that otherwise cannot be directly evaluated. Now, in part, this is because we can look at the sub-processes, many and complex though those might be, that underpin these behaviors that we can look at, as it were, from the surface, right? We can open the hood. We can start to look underneath. Remember that something even as simple as reading a single word out loud involves all of those many distinct sub-processes. If we care about how these complex behaviors can be taught, about how they might go wrong, or how they can be remediated, then it will help us to develop methods like these that permit us to look at those sub-processes in detail. If we can look at those brain activation signatures, then we can start to disambiguate questions about effective methods for teaching and remediation across the lifespan at an appropriate level of detail. Now, in order to meet these goals, we work in a very multidisciplinary framework. I'm, as Provost James mentioned, a neuroscientist, a linguist, a speech-language pathologist, a mom. That's the hardest. My team is similarly multidisciplinary. We work with teachers and educators from across the lifespan, policymakers, psychologists, clinicians, educators, musicians, mathematicians, as well as the engineers and programmers and statisticians and the other experts who make this kind of work possible. Neuroscience is multidisciplinary at its core, and that is a keystone for our research program. And finally, and perhaps the point in which I take the most pride, we don't carry out neuroscientific investigations and then spend time trying to figure out how our findings could be applied to education or to clinical work. On the contrary, our work begins and ends with the translation. Since we come from these varied backgrounds, we bring with us experiences and concerns that we want to elucidate through neuroscience for the benefit of others. So our investigations are conceived and designed with the intent to inform clinical or pedagogical approaches and to shed light on issues of social justice and equality. You know, even though we all strive to make sure that educational resources are available for all sectors of the community, we know that there are inequities in terms of who can access those resources. Through the kinds of research that we do, we're aiming for a clearer understanding of the reasons for those inequities, the barriers that people might experience when they're trying to access those resources. We're looking for more effective means to evaluate underlying mechanisms and an evidence base that will permit us to derive policy changes, changes in programming. We want to equip educators with the tools and the evidence that they need in order to be more effective, to implement the best practices in their fields, to be both producers and critical consumers of the research. We want to find new ways to apply what we've learned about learning. We want to find better ways to evaluate how effective our approaches are, using the most rigorous and objective means that we have access to. And this, I think, right there, that's the promise and the potential for educational neuroscience. One is that when we're talking about mental models within this paradigm, we're not talking about the same kinds of mental models that you're talking about. So what we're talking about is a different kind of reasoning approach where you use visual information to build a model of a situation in your mind, which you then can manipulate in order to solve the problem, right? So, well, no, not exactly, forgive me, not exactly. What you're talking about is perceived and learned experiences that people then use to inform their attitudes and their approaches. Correct. Now, our research actually addresses these kinds of issues, but in slightly different ways. So let me tell you about some of the language-based research that we did, not to get too far off track, but hopefully it will make sense if I can just provide the framework. We've been looking a lot at situations, complex socio-linguistic situations that we refer to as diglossic situations. And diglossia is a context where varieties of a language are separated by their socio-linguistic function. So you have a high variety and something that's referred to as a low variety of a language. The situation that we first investigated using these modalities was actually Arabic, where you have standard Arabic and then you have colloquial Arabic. And colloquial Arabic is the form of the language that's spoken within the family environment, but you have to acquire standard Arabic to be able to participate in education, in literacy, in any form of formal interaction, government, commerce, and so on. The perceived approach to that socio-linguistic situation is that there's really only one variety of the language that matters, and it's the high variety. But using our brain imaging approaches, we were able to show that, in fact, these two varieties of the language are really processed the same, right? They're both languages. They are both represented in the brain in very similar ways. They have the same weight of representation. They are both languages. It's not the case that one is a language and one is a dialect, or that one is a language and then you change register, right? Like you speak a poorer version of that language or something like this. That's not what we found. We were able to show that actually, the representations and the processes are equivalent between these two. And that to us was very interesting, because in Arabic you have a situation where people are speaking, children are speaking one language at home, then they get to school and they're being tested in a different language, right? So actually a different language. It's like, if I raise my child speaking English at home and then he goes to school and he has to be tested in Spanish, right? It's that equivalent. Our extension on that work, and something that we've actually just begun, well, actually that led to a couple of things, because we now, with my colleague, Professor Khamis Dakwar, is a native speaker of Arabic, and so we've started to develop new ways of assessing language that take account of the diglossic situation, right? So that we're not actually asking people to test in one language when they speak a different language. So that's one way in which we're starting to implement some of these different policy and programming approaches. But a new piece of research that we're trying to do is to identify, what other situations might that be happening that we're not recognizing. And so we're starting now a program to look at African American vernacular English, right, where children are speaking one variety of the language at home, and then they get to school and they have to be evaluated in some other variety. Now, that work it's new and it's in progress. We don't yet have findings that we can report, which is why it didn't make it into tonight's presentation. But those are the kinds of ways that we can use these techniques to help us, disentangle the social and cultural aspects of these questions and really look at, is that right? These perceptions that we have, these things that we think we know about language, that we know about culture, is that really what we're doing at the level of the brain? Is that what's happening? And in fact, we can answer those questions in different ways now.
I wonder if there are any other aspects that you're looking at practice over time that can make...
Well, we're looking at meditation. Shall I address that? There's, a lot of the work that we do actually, looking at it longitudinally would be extremely valuable, and that would be, the next step for a lot of this work. So the MMNs in childhood apraxia, for instance. So if we look at children who don't have MMNs now, but we look at them over the course of development, do we see that changing? We need to kind of follow it through. We look at how brains respond to languages they've never been exposed to before. And then we follow people through the learning experience, and we see how those responses become more organized over time, right? So those kinds of approaches, yes, practice is something that's really crucial. Exposure is really crucial. Practice in terms of meditation, so shall I talk about that? It's actually really kind of cool, but again, it's something that we just started to do. So, it didn't make it into the presentation tonight, but it is something very interesting. When I was working at MIT, I was involved in a project looking at language processing in schizophrenia. And I worked closely with a group of psychiatrists from McLean Hospital. It was a collaborative program. And we identified a group of adults who had schizophrenia, but who were stable and well managed, so they can participate in brain imaging experiments, right? I mean, this is not something easy for people to do. And we looked at their brain function as they were carrying out a language processing task that they could do well, right? I promise I'm getting to the point. So they performed on this language task the same way as our control group, right? They were just as good at the task, just as quick at the task. But when we looked at the brain responses, we saw extreme disorganization of the brain processes that were involved in this, right? So especially compared to the control group, where you really see like a very clear sequence of events, that sequencing was not apparent in the group of people with schizophrenia. And this kind of disorganization, it led me to think, how can you address that? If you're seeing really disorganized brain responses, and yet the behavior is being managed, then somehow the brain is handling a situation, right? But is there a way that you can you change the way that the brain is responding? Can you alter, make it more organized? And I did, that was something I worked on then, and I didn't get to address it again for a number of years. And a couple of years, 3-4 years ago, it's funny how fast the time goes, had an opportunity to have some Buddhist monks visit my lab. And so you get Buddhist monks in your lab, you're going to put electrodes on their heads, right? They were very gracious and understanding about it. So we had a very simple kind of sensory processing experiment, and what we did was we ran that experiment before they meditated, then while they were meditating, and then again after meditation. And we had 3 monks, and 2 of them were younger and more junior monks. They'd only been meditating for, 20 years or something. So, and what we saw with them was that, their brain responses to the sensory processing looked very much like, yours or mine, right? Sort of typical. And then when they meditated, I saw this change in the coherence of the brain activity, that it became much more organized, right? And I'm like, ding, ding, ding. And then after they meditated, we saw that some of that coherence stuck around, but it got less, right? So there's an effect afterwards as well. And then we had the senior monk, the Rinpoche, who had been meditating for like 50 or 60 years. And you know these people, sometimes you meet them, he was like glowing, you know, like that. And we put the electrodes on him and asked him to, you know, sit in the chamber, and we plugged him into our equipment. And I thought my equipment was broken. I'm not kidding, because everything was thoroughly organized and beautifully coherent from the beginning, before he started meditating. And so, looking at this, I really did unplug everything, like shut it all down, start it all up again, you know, like just checking, like really, is this, you know, put it on me, okay, no, that's the same as usual. And, you know, when we asked him, you know, you weren't meditating then, right? And he said, of course. And so we said, but, you know, we need to do it before you meditate and then during meditate. And he said, but I'm meditating right now. I'm meditating when I'm speaking to you. I'm meditating. And this kind of organization of brain activation, you know, it did lead me to think that, yes, it's clear that over many years of practice, you see differences, you see changes. What we don't know is how long it takes to induce those changes. And so that's work actually that we'll be beginning. We also don't know what are the effects of that kind of practice on pathological situations. So we're not going to begin that kind of work with people who have schizophrenia. It's a very vulnerable population. You know, that there's a lot of work to do before we can justify applying it in those situations. TC has this mindfulness and education working group, and so we've been talking to those people. They actually go to the speech pathology clinic and they work with the stroke group. And they provide some, you know, basic meditative practice for people post-stroke. And so we've been talking to them about, in the future, we have a lot of work to do first, right? But in the future, looking at how meditation practice could affect the brain responses of people post-stroke, for instance. And it seems to me like that would be an interesting, you know, first couple of steps towards trying to think about how we might support people who do show those kinds of extreme disorganization of brain responses.
Any other practices, like aesthetic practices? Are you doing any research in that, or doing any research?
Of aesthetic practices, such as
music education.
And dance, for instance. So not being a musician or a dancer myself, I have actually had opportunities to tap into the expertise with respect to that. A couple of years ago we did do an experiment looking at the brain responses of expert musicians versus people like myself who you would not describe as an expert musician. When they perceive, it was kind of cool, when they listen to sort of small tunes, and then you violate the expectation of how the tune will end. You know, you make something that's in a major key go minor, or something in a minor key go major, or you just give it like the wrong ending. And even though everyone can perceive that, you know, when something is wrong, like everyone, even I can tell, you know, that it's not right. But what we see in the brains of expert musicians is a much more rapid, early response to those kinds of violations. So it's something more kind of rule-governed. Whereas for people who are non-expert musicians, even though they're perceiving it, they're processing it in a very different way. So there are those kinds of approaches. We have somebody at the moment who's interested in looking at dancers and how they use mental imagery. But these kinds of approaches, you know, they're very difficult to operationalize for experiments. Like, it's hard to get somebody to sit there with electrodes on their head, not move too much either, right? So you can't, like, because if you're moving, you saw what happened when Heather nodded her head, right? So if we've got somebody actually dancing or actually playing an instrument, then we've got all kinds of problems there. So getting people to use mental imagery and then stop using mental imagery for a second, okay, now start again, right? And now stop. Like, experimentally, it brings challenges with it, but we certainly are thinking about those kinds of approaches, yes.
Going back to your N170
Sure.
responses, I was wondering how that might look or apply to people with dyslexia.
Absolutely, yes. I think that's an extremely valid question. We do have a dyslexia study in the planning stages at the moment. And I mean, we see changes in the way that people process that. We're trying to identify, before we start looking at reading in dyslexia, and this is the problem with talking to a science person about this, right? Because we get down into the details and everything kind of goes back and back. But before we actually get at reading in dyslexia. We're trying to evaluate visual processing in dyslexia to see like how far down the chain do we have to go before we stop seeing differences, right? So that's actually where our research is at right now. We're looking at perception of motion and color in people who have reading disabilities. And when we've done that work, then we can start to look at, well, you know, like what's the reading effect. For sure, we will see differences, yes. And there's already a substantial literature looking at it.
I'm wondering, with all the different facets of your research, if you've ever done any research into colored language synesthesia and people who experience that.
The short answer is no. Synesthesia is such a fascinating situation. Most of the work that we're doing at the moment, okay, so there's sort of a big picture way to explain this, right? That what we're trying to establish at the moment are the ways that brains do stuff. And that means we have to look at how everybody does things the same. Because once we understand that, then we can start to look at, you know, what are the differences? What things can change? And that's when we could really get into something like synesthesia, which is, it's kind of like when the modular organization of cognitive processing breaks down and you get this sort of blurring between the different sensory domains. I mean, I think it's really fascinating, but we haven't got there yet. Yeah.
Find out what they're doing. They are here, and we're going to open the doors now and enjoy. Thank you.